My name is Vera Kalscheuer. I'm a group leader at the Max Planck Institute for Molecular Genetics in Berlin. And I'm interested in developmental disorders or disorders with intellectual disability since the starting with my postdoctoral research. And today I will present the molecular genetic spectrum of CLCN4 related neurodevelopmental condition. And I will include unpublished results that we have obtained with the support of many families and through many international collaborations over the last years. How did I come to study okay, you don't see the upper part, but it doesn't matter. How did I come to study CLCN4? Among various other activities, my group has spent more than 20 years on finding novel disease genes for intellectual disability or neurodevelopmental disorders, to help families with a genetic diagnosis, to learn more about the clinical phenotypes, to learn more about the gene function and the protein functions, and also to learn more about the pathogenic mechanisms. And for many years, the focus of these studies has been on the X chromosome. And this slide shows you what we have achieved during this almost 20 years, either within my group or in collaboration with many other groups. And this, of course, can't be without large international collaborations. To date, there have been 24 published families reported in 7 publications. And while the first report by Vera and colleague was on a single case, a boy who carried a de novo variant in CLCN4 and supported by functional studies. This was the first report showing that CLCN4 is a novel candidate disease gene. Soon afterwards, we performed a large study on families, on 405 families with who did not have a genetic diagnosis at that time. And those families had, from the pedigrees of these families, we assumed that those would have a variant in one of the X chromosomal genes. And in these through the studies, we identified 7 novel intellectual disability genes, and one was CLCN4. For CLCN4, we identified 4 missense mutations and 4 independent families, and also 1 truncating variant in 1 family. Soon afterwards, we reported on 11 additional families who carried de novo and inherited variants in CLCN4. And in this report, we described for the first time on affected females who carried a de novo variant in CLCN4. Also in 2018, Sue and I reported on a male who carried a recurrent de novo variant. And last year in 2021, there were 3 reports on 6 novel families who carried either a recurrent variant in CLCN4 or novel variants in CN4. In human genetics, as many of you know, the interpretation of missense variants can really be challenging. And therefore, we continued our studies. And this slide shows you an overview of the published variants in CLCN4 along the gene. And this is the one dimension of the protein with the various protein domains. And these are the published variants up to now. There are 2 truncating variants, as I mentioned before. We see one in frame deletion, and we also have a splicing variant. Those boxes shown in pink, open boxes in pink, denote de novo missense variants in females, and open boxes with blue color show de novo missense variants in males, whereas filled boxes indicate inherited missense variants in males. And this one is an inherited missense variant in a female. So shortly after our publication was online in 2016, we were approached by many clinicians who sought advice on how to interpret the newly identified CLC4 variant. So then Emma and I shared our opinions and found that we were both keen to continue our studies on CLC4 to help families and to learn more about the CLC4 condition. And we were both very much delighted that Michael Pusch, who will speak just after me, shared our opinion and joined us. And I call this a dream team. So what did we do? We assembled a database of variants identified in CLC4 with information from clinicians and diagnostic labs who contacted us. And we also contacted the labs or clinicians who deposited CLC4 variants in databases, for example, ClinVar, which means Clinical Variation Database. And we also included variants published by others when recurrent with our entries. So in total, at the moment, in this database, we have 79 missense variants, 48 are unique and 11 are recurrent in 31 families, and also 3 novel truncating variants. Those variants were identified by either gene panel sequencing, exome sequencing, or by singleton or trioexome sequencing, whole genome sequencing. And the laboratory assessments on pathogenicity following American College of Genetic Guidelines said 1 is benign, 42 are variants of uncertain significance, 23 are likely pathogenic, and 6 are pathogenic. And although we have tools to predict the pathogenicity of missense variants, it's always a challenge also for families if they get a report that the variant identified in the affected is of uncertain significance. Also in this database, 30% of the variants had at least one prediction tolerated benign or low scores, which means that they also could be very rare polymorphisms. And a few variants, 14, were also present in population databases, which means that they are either very, very, very rare or polymorphisms, or that, here you see the frequency in heterozygotes, which doesn't mean that one or the other of these variants can be pathogenic in boys or in males. And so we decided to study 59 of the CLC4 missense variants in total in xenopus oocytes, but of the 25 variants that had at least one low prediction, 12 were functionally like wild-type CLC4, and 13 of the variants showed a functional effect. So from now on, I will present the molecular genetic spectrum of 43 unpublished families for which we have been able to gather clinical information and also segregation data. So this shows you an overview of the 25 families with affected males. 16 of the males had carried a missense variant, and also we identified 2 truncating variants inherited in males. These are shown here. And 5 missense variants were de novo, and 1 of the truncating variants was de novo. And for 1 variant, we don't know. And this gives you an overview of the 20 families with affected females, who all carried a missense variant. And again, 5 of the missense variants were inherited in females. These are the ones with the colored squares. And I also would like to point out here that 1 of the missense variants in this affected female was inherited from a mildly affected father, so paternally inherited. And 15 of the missense variants identified in males were de novo. So this shows you an overview of all the published variants and newly identified variants. And we see that the numbers increase. So we have 30 inherited variants in males, 12 de novo in males, and 5 inherited in females, and 20 de novo in females. And as you will hear in the next talk by Michael, those which are highlighted by a thicker line, for those in xenopus oocytes, Michael has found that those are gain of function variants. Oops. And here I want to highlight that with more and more variants being identified, we also see CLCN4 variants which are recurrent in families, and for which we also do have functional results. So this, for example, means that newly identified variants in other families or recurrent variants identified in other families will be more likely to be judged or interpreted as pathogenic instead of variant of uncertain significance. So what did we find? Everything you can imagine in human genetics. The CLCN4 variants can either be de novo, maternally inherited, or paternally inherited. In a few individuals, we also saw that the variants are mosaic, so only in a small number of their cells. And up to now, the molecular genetic spectrum of the affected from 68 families, which means 24 published and the 43 unpublished, for which we have clinical information. We have 90% of the variants are missense variants, 5 truncating variants, 1 small in-frame deletion, and 1 splicing variant. And of note, 80% of the variants carried by affected females are de novo, whereas 27% of the variants carried by affected males are de novo. And in total, we have 5 recurrent variants in 15 families. However, if we include entries from ClinVar database, which come in almost daily, and most of them without clinical information, to date, we have 14 recurrent variants in 39 families. And in silico predictions do not always match with functional outcomes. It's clear that further studies are needed to understand what CLC4 normally does and what goes wrong in CLC4 deficient cells to cause the symptoms of the disorder. And if we know what went wrong, we could find starting points for developing treatments. And to do this, we need to find out what CLC4 does in our cells under normal condition to keep them healthy. So this is my last slide. I would like to acknowledge all the patients and the families without whom those studies wouldn't be possible. I would like to thank Vanessa Zucco and Jessica Gibbons from my group. Special thank to Emma Palmer, who will also speak right after Michael, from Sydney Children's Hospital Network and University of New South Wales with her colleagues. And of course, a special thank to Michael Pusch from the Institute of Biophysics in Genova and his team. And as you can see here, many, many more clinicians and others who have contributed to this study. Thank you for your attention.